Hello again, and today we are going to start our final section in the Fundamentals of Pharmacology course, and it's going to be a direct follow-on from the preclinical drug development piece that we talked about in the last class. So this is section 11, drug development, part 3, and we're going to focus on human drug development, otherwise referred to as clinical drug development. So we've got several objectives for this particular class that include the following. We'd like you to be able to understand the 4 distinct stages of drug development. We'd like you to be able to define the purpose or purposes of each of these stages of development. We'd like you to have a sense of the structure of the study protocol, the research protocol for drug development, and the similarity in this structure across all stages of drug development. Ideally, what I really want you to be able to do is to apply your knowledge of pharmacokinetics and pharmacodynamics that we have taught throughout this course to the drug development process, and how your knowledge of those concepts are key to understanding and appreciating the complexity of the drug development process. Finally, I think it's incredibly important that everyone appreciates the level of oversight that exists during the drug development process and how regimented and regulated this process actually is. So there are 5 objectives, and I hope I'm going to be able to get all these across to you within today's class. So it is a total understatement to say that drug development is highly regimented. It really makes no difference what country you're in. Every nation has its own regulatory processes that govern the process of drug development at every stage. And no matter what country you're in, there are 3 basic principles. It requires extensive planning. It's heavily regulated by the government, and it is unbelievably costly and time-consuming. So the regulatory oversight is going to differ slightly between countries, and I'm going to use my reference point, which is drug development within the US. So prior to actually taking a molecule from your animal studies or your overall preclinical studies into a human for the first time, there has to be a regulatory checkbox achieved. And we call this an approval in parenthesis because essentially it's a permission to enter into human testing. And it's not that the regulatory authority is approving the drug in any manner, shape, or form. They're just providing a green light to go forward to test the drug in humans in the experimental setting. In the US, this is earmarked by obtaining an investigational new drug number from the FDA, and there are going to be similar structures in other nations. And this obtaining of an IND, investigational new drug, is required prior to giving a new drug to a human for the first time. And we call these, not too surprisingly, first-in-human studies. Or it is also required in many circumstances if there is a plan to study a drug that's already been approved, but you want to study it for a new indication. The process involved here involves layers upon layers of oversight and is unbelievably complex. For example, in the US, it would be the FDA who provides the IND. In Europe, it will be the EMA. In Australia, South America, etc., you're going to have different regulatory bodies. In addition, much of the research that's done in academic institutions in the US is funded by the National Institutes of Health. This is a national federal funding agency. There is an ethics board approval that is also required in the US. These are called institutional review boards. They're typically referred to as ethics panels or ethics boards in other countries. The management of the trial, the actual conduct of the trial, frequently employs the services of an external entity called a clinical research organization, or a CRO for short. This is a very large body of individuals who provide monitoring and regulatory oversight for studies that are conducted within the US, particularly within academic medical centers or within hospitals or health systems. Then there's the sponsor, and the sponsor is typically the pharma company that is providing the drug for the investigation. And I say typically because it's not exclusively that scenario. The investigator and the research team. So the concept here is that everybody works in harmony, that everybody adheres to what the regulations that are required for each step in the drug development process. It is the sort of undertaking that requires a degree of training. We can't just go in and conduct clinical trials without having a knowledge of the regulation and the oversight that is necessary for ensuring that these studies are done correctly. And the primary purpose of regulation is such that the data that is obtained from the clinical trials involving humans is robust and reasonably infallible. The idea that the data that will ultimately be examined by the FDA or the regulatory authority in your own country to ultimately approve the drug for use in humans has to be strong, reproducible data. So this level of oversight is in place to protect the subjects who are participating in the research studies, but also to ensure that the study is conducted appropriately and that the data that's obtained from the conduction of the study is going to be usable in such a way that the drug may ultimately have value for the human population in general. Now, there is no intention whatsoever for you to be able to read every line on this slide. I'm just giving you a semblance or a sense of the spectrum of legislation that pertains to the regulation of drugs just in this country, just in the US. And the FDA is the cornerstone of regulation concerning drug use, drug approval, drug monitoring within the US. So very complex legislation which is ever evolving. So what are the phases of clinical drug development? Also, as I said previously, human drug development. Well, we refer to them as phases. So phase 1, phase 2, phase 3, and phase 4. Phase 1 studies, the primary purpose are to assess the safety and the tolerability. Phase 2 studies, the primary purpose is to try and identify an appropriate dose that brings about an effect. Phase 3 is looking at something called the therapeutic ratio, that the drug, when it's used in clinical practice, actually improves the clinical condition, either reduces the rate of progression of the disease, prevents mortality or morbidity, another word for saying illness, morbidity from the illness, or cures the disease completely. Phase 4 studies are referred to as post-marketing surveillance studies, and these studies are conducted after the drug has been approved by the FDA and is being used in clinical practice according to the FDA approval guidelines. So returning to phase 1, the safety and the tolerability is primarily focusing on pharmacokinetic principles and early pharmacodynamic principles, particularly with respect to tolerability. The phase 2 studies invoke the principles of pharmacodynamics primarily, but they continue to evaluate safety. And it's important for me to mention at this stage that every phase of drug evaluation includes a safety assessment. And I'd like you to remember that point. Every phase of drug evaluation, both before and after drug approval, involves a safety evaluation. The therapeutic ratio is different from the dose finding stage of drug development in that the dose finding is looking primarily on a surrogate marker, and I'll come back and explain exactly what a surrogate marker is in 1 second. So this is a high-level overview of the 4 stages of clinical drug development. Now you can imagine that if regulatory authorities around the world are reviewing the decision whether to approve a drug for use in clinical practice, it's very, very helpful if the design and the conduct of clinical trials is uniform. And that's exactly what's in place. There is a uniform process whereby clinical trials are conducted, not only in terms of phase 1, phase 2, phase 3, and phase 4 being conducted sequentially, but also the construct of the clinical trial or the phase of drug development is exactly the same. And all of the phases of drug development, we can interplay the term clinical trial. So we can have a phase 1 clinical trial, a phase 2 clinical trial. So each phase involves a clinical trial. And what I'm attempting to show you on this slide is the headers, the headlines for each of the individual sections of a clinical trial protocol, a study protocol. So each of these, irrespective of the phase of drug development that is underway, will include a background section, a primary hypothesis, a primary endpoint, the efficacy and safety assessments that are intended, the study population, the control conditions, the method of randomization, the blinding conditions, statistical analysis plan, quality assurance measures, data management, and study oversight. And I will go through each of these in the next few slides. So, first of all, background. The primary purpose of the background section in any clinical trial protocol is to provide a justification for why the research team or the pharma company wants to undertake that phase of clinical drug development. So, for example, in a phase 1 study, the background section would be detailing the data from the drug discovery phase and the preclinical drug development phase. So it might discuss issues such as target of drug action, mechanism of metabolism, route of elimination, all of which have been calculated from animal model systems. This data would be included in the background, sometimes also called preliminary data section. In addition, in this section, in the background section, is included a rationale for why it is important to evaluate this drug to improve the management of the clinical condition that the drug is intended to be used in. So, for example, if we were developing a drug for the treatment of hypertension, the background section would include an initial paragraph to say that more than 55 million Americans have a diagnosis of hypertension, and the current control rate is 30%. Therefore, there is the unmet clinical need to develop novel therapeutics for the management of hypertension. The background section would then go on to say, we have a novel compound, drug X, which in the drug discovery phase appears to block a receptor that's on the cell membrane that participates in the generation of elevated blood pressure. In addition, this molecule has been demonstrated to be both efficacious as well as tolerable in the following animal species, and would then go along to depict what those animal species is. So you can appreciate that the background section, which includes all of this information, might in fact be quite lengthy in a phase 1 trial. Then imagine the background section for the phase 2 study, because now when you get into the phase 2 study, not only are you going to include the information that was in the background section for the phase 1 study, but now you're going to add in the data from the phase 1 study to provide further justification for continuing into the phase 2 study. And then if you're conducting a phase 3 study, you would include the phase 1 as well as the phase 2 data in the background section of the phase 3 clinical trial. So that's the background piece. It can be very lengthy depending on the nature of the research that is about to be undertaken. The next setting is the hypothesis, and I appreciate that many of you out there may have no prior experience with research. And hypothesis is basically a best estimate of what you predict the outcome is going to be. So when you think of hypotheses that might exist for a phase 1 and a phase 2 and a phase 3 study, they're going to look something like this. And what I want you to do is keep in mind, primary purpose of phase 1 is safety. Primary purpose of phase 2 is pharmacodynamic effect. Primary purpose of phase 3 is efficacy on clinical outcome. So a phase 1 primary hypothesis might read something like this. Drug XX001 can be safely administered to humans. Now, obviously, it's not going to be as simplistic as this because safely needs to be defined. Safe in terms of kidney function, renal function, lung function, cardiac function. So pretty broad spectrum. Pharmacokinetic principles. Phase 2 is going to read something like drug XX001 dose-dependently inhibits whatever your measurement is. Now remember dose response curves? This is your phase 2 development piece. Now you will have conducted a dose response analysis in the animal studies. So that is going to inform how you're going to conduct your phase 2 study in humans. So let's stick to our example of an antihypertensive drug. The phase 2 study would be my antihypertensive drug X dose-dependently lowers blood pressure. That's a surrogate marker. Another example might be in the field of cholesterol management. Drug X dose-dependently lowers LDL cholesterol. Or it might be a drug for the treatment of cancer. Drug X dose-dependently inhibits a marker of cancer biology. CEA 125, for example, is a marker of ovarian cancer. Or in the HIV world, drug X dose-dependently lowers CD4 counts. So these examples are all concepts of how a drug positively impacts a surrogate marker for disease outcome. Phase 3, in contrast, would be something like, as compared to whatever your standard of care might be, drug X decreases mortality. So let me use your blood pressure example again. We know that we use blood pressure medications to reduce cardiovascular events, to reduce stroke, and to reduce progression to renal failure. However, we use blood pressure measurement as our surrogate marker for how effective these medications in improving mortality and morbidity. So the phase 3 clinical trial hypothesis would be, compared to a standard of care, let's say a water pill, drug X decreases mortality by virtue of lowering blood pressure. So decreases mortality is your clinical endpoint. So these are your hypotheses. And you will note that when you state your hypothesis, it also defines what's called the response variable. And that simply means what you're going to measure. So in your phase 1 study, it might be liver function tests. It might be white cell count. It might be alteration in pathology staining. It might be measurement of metabolic enzymes. In a phase 2 study, the response variable, the example that I gave you, was blood pressure lowering, CD4 count, tumor markers in the case of cancer. In the phase 3 study, it's going to be a clinical event, cancer burden or tumor burden, more commonly stated, stroke, heart attack, death from HIV complications or HIV-associated infections. The hypotheses, stating the hypotheses also gives you the opportunity to ask lots of other questions. However, there's always one central primary hypothesis, even if there are a load of secondary questions and variables that you might want to mention, and don't worry too much about that piece. And as I said previously, the hypotheses will always include a safety hypothesis or a safety guess that you constantly continue to list safety assessment. And you do this by a priori stating you're going to measure the number of adverse effects that occur throughout every single stage or phase of drug development. And what happens then, as you go through clinical trial development or human drug development, you start to see questions being asked of subgroups of populations within larger studies. So sticking to the example of hypertension, you might want to know, do different racial subgroups respond to antihypertensive drugs in different ways? So within your clinical trial, you might have a Caucasian subgroup, a Hispanic subgroup, and a Asian subgroup, African American subgroup, etc. And you want to ask sub-questions or secondary hypotheses. Are there differences between these subgroups? Another example might be age. You might want to study what the impact of a pain medication might be on, say, children between the ages of 1 and 10, adolescents between the ages of 10 and 20, young adults between the ages of 20 and 30, etc., etc. So it allows you to refine your question because you continue to adhere to this very structured skeleton in which the clinical trial is actually conducted. So you have your primary hypothesis, now you convert it into a primary endpoint. And the main reason, or excuse me for saying this, the primary reason for having a primary endpoint is so that you can impose statistical analysis upon it. So it allows you to state your primary purpose, your primary measurement endpoint in such a way that it's definitive. So for a phase 1 study, the primary endpoint would read like to determine the safety of drug X, to determine the pharmacokinetic profile of drug X, and remembering that pharmacokinetics involves administration, absorption, distribution, metabolism, and excretion. It might read something like to determine the maximum tolerated dose, much like what was done in the earlier stages of acute toxicity and subacute toxicity in the preclinical studies. We do exactly the same thing in humans. We want in these early phases of drug development to be able to identify what is that maximum tolerated dose that we might want to lead with when we go into the phase 2 study. And it's important to appreciate these are typically done sequentially as you go through the phases of drug development. And then the primary endpoint for a phase 2 study is going to sound very much like the hypothesis, but stated a little bit less, sounding like guesswork. It's going to say something like to determine the effect of various multiple dosing regimens of drug X on blood pressure, CD4 count, protection against infection, protection against contraception, etc., etc. Phase 2 is also the phase at which we learn what's the least amount of drug we can get away with, minimum effective dose. And you'll remember we talked about that in our last class, MED, minimum effective dose. In clinical medicine, we want to always use the least amount and the lowest dose possible of any medication to bring about the wanted effect of that drug. And the idea being if you do that, you limit the toxicity. Phase 3 primary endpoint is going to read something like to determine the mortality rate of drug X treated patients compared to the mortality rate in Y treated patients. Now, I'm going to introduce you to a concept which we'll extrapolate in a couple of moments, and that is that when you look at clinical endpoints, you nearly always have a control population. And that is because you want to be able to compare outcome between two similar populations so that you're able to say it's by virtue of the intervention, the experimental intervention, as opposed to some environmental cue that created the change. So it allows for an element of control, and we will return to this concept in a couple of moments. So in every clinical trial protocol, you declare upfront what you're going to measure. And these are synonymously called assessments or dependent variables. It's entirely up to you what term you use, assessments or dependent variables. And this is, again, a degree of reiteration of what you will have said in your hypotheses and your primary endpoint. At this stage, you may be scratching your head a little bit and wonder, why are we so repetitive? Well, one of the reasons is because You want to be definitive. You want there to be no ambiguity about exactly what you're doing when you expose humans to clinical trial development. So you say up front what you think is going to happen, you then state what your endpoints are going to be, and then again you state what your dependent variables are. And typically the dependent variables and the endpoints are going to overlap. So your phase 1 endpoints, safety, pharmacokinetics. Say phase 2 endpoints, continued safety, please remember that. Pharmacodynamic endpoints, i.e. surrogate markers. Phase 3 endpoints, clinical endpoints, and you can also continue to measure your surrogate markers. This is actually quite common, to continue to measure surrogate markers, because in essence, what that allows you to do is to continue to solidify your belief that your surrogate marker is indeed a robust surrogate marker for your clinical outcome. In other words, in the HIV world, that CD4 counts CD4 counts are indeed a reliable surrogate marker for HIV clinical endpoints. Blood pressure is a solid reproducible marker for stroke endpoints or heart attack endpoints. So continuing to measure surrogate markers in your phase 3 clinical trial enables you to garner even more information than just clinical endpoints. And when you think about how expensive these studies are, it makes sense to garner as much information as you possibly can at every stage of drug development. So far, we've checkboxed background, we've checkboxed hypothesis, primary endpoint, and safety assessment or as dependent variables. So now move on and let's talk about study population. What does this term mean? Essentially, it means who you want to enroll into your study. What is the best population in order for you to be able to answer your primary hypothesis, your primary question? Well, returning to phase 1, remember that in phase 1, we're looking at safety. Typically, and I'll emphasize typically, these studies are done in healthy people. So the age range by default tends to be between 18 and 40. For ethical reasons, we do not typically enroll children into phase 1 studies unless the primary indication for the drug is going to be in children. So the common age range for phase 1 studies is the healthy 18 to 40-year age group. We try to enroll equal numbers of men and women. With respect to women, we will enroll women with no childbearing potential. The primary reason for this is that we want to have a very good sense of safety before we would run the risk of a participant becoming pregnant and being exposed to a heretofore unknown adverse toxicity profile. These studies are typically small. N means the sample size, the number of subjects, usually around 10 or 20. So these are small control studies. They tend to be what we call pure populations, so without any concomitant diseases and on no other medications. There are exceptions to this rule. For example, if we have a very toxic drug that we know is a very toxic drug, well, then the therapeutic index of that medication becomes very, very important, because we would only expose patients who have that risk-benefit analysis of being exposed to a toxicity that is acceptable. So let me give you an example of a chemotherapeutic drug. We have different types of study designs. Phase 2, the study population is going to be a little bit broader. So here what you want to do is actually choose patients with the condition which you intend this drug to be used to treat. So here you're going to have a wider age range, so 18 to 70 is pretty representative of the age range for most phase 2 clinical trials, and you want them to be a fairly low-risk patient population. So let's say a stage 1 or stage 2 hypertensive patient who doesn't have much in the way of comorbidity. They don't have known cardiovascular disease. So considerably lower risk than somebody with poorly controlled hypertension and a prior stroke, for example. We also enroll men and women. Again, we try to keep the risk of teratogenicity in humans to the bare minimum. So we're very careful about contraception protocols to try and prevent any unwanted or unintended pregnancies occurring. In the phase 2 studies, we want to see what the interaction of the medication is going to be in the setting of other essential, commonly used medications. So let me give you an example of, let's say, a 58-year-old diabetic, hypertensive, hypercholesterolemic female. And they are entering into the study, into a clinical trial, phase 2 clinical trial, to evaluate a new blood pressure lowering pill. In this sort of example, it's very important for us to start to develop information about how the experimental blood pressure lowering pill behaves in the setting of other medications, say for treating her diabetes or treating her high cholesterol level, because these are the sorts of concomitant medications that are going to be seen in clinical practice were this drug to be approved. So some essential concomitant background medications is usually the norm. In terms of the study designs themselves, they can be short-term multiple doses, they can be longer-term doses. Again, it depends on how you intend to use the drug in clinical practice.